Hi everyone, Eddie's here. Today we're going to learn how we can use ImageJ to analyze some images. The first thing you need to do is to open up ImageJ, and you should already have it installed. The second thing you need to do is to find where exactly you saved your image. And remember that we saved the images in the proprietary format of the system that we used. Okay, so ImageJ has a special collection of plugins that allows us to open all of those extensions. So let's open this file in ImageJ. As you pull the file into ImageJ, it's going to show you a window that looks like this. And for now, we're okay with open them as a hyperstack. So just click on OK, and it's going to open the image right over here. Now, what do we see here? So down here, you see a slider. If you move the slider, it has three images. These three images are the three separate channels that we acquired in the lab. Now that you will note that the image that I'm using is a little bit blurry, and that's because I didn't clean the slides before the cover slips before imaging. But again, the focus of this tutorial is to learn how to analyze, and we will learn how to take beautiful images whenever we are in the lab. So again, we have three channels: the first channel, the second channel, and the third channel. What are these channels? Well, remember that when we acquired these images in the lab, the first channel was our DAPI, the second channel was our GFP, and the third channel was our phalloidin stain. So this is actin. And we are looking here at the HeLa cells that have been transfected with GFP and stained with phalloidin. Okay, and ND2 is the specific format that Nikon system uses. So what do we know about this image? Now, because we saved this file in this format ND2, we have all of the metadata that's associated with this file. So if you go over here to Image and Show Info, this will give you a lot of information about the system that acquired this image and how exactly this image was acquired. So for example, it will tell you what kind of objective you used, what kind of binning you used, what kind of exposures you used. All of this information is saved in here. Okay, so this is very, very useful. It will also tell you the name of the camera that was used and all this information about the system itself. So make sure you scroll down this thing and see what kind of information you can find here. Now let's see how we analyze the image then. The first thing that we note from this image is that it is black and white. There is no color here. This is a little boring, so we want to add color. We are going to add blue, green, and red colors. There are two ways of adding colors in ImageJ. The first one is using lookup tables, and the second one is by splitting the images and merging them, and I'll show you both of these methods. So let's start with the lookup tables. So remember that the camera, when it acquired these images, it acquired them as black and white images. So there is no color associated with the image. So we can use these lookup tables that you can find on the image, and right here, lookup tables, to add some color to it. And you can see some different colors, and I encourage you to play with all of them and see which one you like the most. For now, I'm going to add the blue color for DAPI. So we just click on blue, and now all of the DAPIs are blue. If you take the slider, you move it to the next frame. Unfortunately, it colored it blue as well. But once again, we're going to go to lookup table, and then we're going to associate the green color with it. So now the system knows that the first channel is blue, the second one is green. And now we have to do the same exercise with the third channel right here. So we know that this is supposed to be red, or we want it to be red, so we're going to associate the red color with it. Okay, so now we have all of these three colors that are associated with these images. Now, what about the other method? Remember I told you that we can split the images and then merge them. So we have these three channels. We can go to Image, Color, and Split Channels. What this is going to do, it's going to break this file into three. The file that contains the blue channel, a file that contains the green channel, and a file that contains the third red channel. So you click on it, and you get these three. channels over here. Now pay attention to the name of each file. C1 stands for channel 1, C2 stands for channel 2, and C3 stands for channel 3. Okay, just remember that for now. Now the reason that they're all colored at the moment is because we just colored them in the previous image. If you were to do this on a file that you just imported into ImageJ, you would actually not see any color. So how do we form a merged image, for example? Or how can we use the merge feature to add some color to these images? So you can go to Image, Color, and then Merge Channels. And this little window will ask you which image do you want to be, red, green, or blue. Or you can of course use cyan, magenta, and yellow as well. So let's say we want this image, the one that's called C1, we know this is Dabi, we want this one to be blue. So ignore this C3. This C3 doesn't mean that this is the third channel that we used. This is something that the software called it. But we want this file, the C1, to be actually blue. So we have to check that this is C1 over here. Blue is going to be C1. Green is going to be C2, that's fine. And red is going to be C3, as we see over here. Okay, and now you click on OK. Now before you click on OK, you can actually click on this as well, and this will keep the source images opened. This is useful at times as well. Okay, and we can actually ignore the original lookup tables. We don't need this because it will add the color that we associated here with each image. You click on OK, and you get this beautiful image. Okay, so now basically you get the composite image. What are you going to do with the composite image? Well, the first thing we need to do is we need to actually save it. When you work with images, it's best to save it in TIFF format. This will give you the highest resolution. For now, however, I'm going to save it as JPEG. So I'm going to go to File, Save As, and I'm going to save it as JPEG. I highly encourage you to save it as a TIFF though. So now it's going to ask me where I want to save it, and I'm going to tell it that I want to save it in this folder as composite. So now this image has been saved as a JPEG image. Now one important thing to remember is that when you close or when you modify the file in any way in ImageJ, there's no really an effective way of going back. So for example, I closed those individual images with the individual channels, and I can't really go back to them anymore. So now this is all I have open. So if I want to get those individual images once again, I actually have to go back and repeat the entire process. Okay, and this is what I'm going to do now. So let me close this file. Let me reopen this one. Okay, and now I'm going to split them into 3 independent channels. But before I do it, I'll just rename them. Well, I'll associate some color with them, sorry. So lookup table, this is going to be my blue channel. This is going to be my green channel, and this is going to be my red channel. Sorry, right down here. Now I'm going to split it. Sorry, right here, color, split channels, and I have these 3 channels. I'm going to save them individually now. So I'm going to go to the blue one, and I'm going to do save as. Again, TIFF is better, but I'm going to save it as JPEG for now. So this is my C1, this is my blue channel, and now I can close it. This is going to be my green channel, so I'm going to save it once again as my green channel, and I'm going to close it, and then I'm going to save this as my red channel, and then I'm going to close this window. So what do we do next? One of the best ways to present this images is by putting them side by side, and you can use multiple tools to arrange your images. Now, because all of you have PowerPoint, I decided I'm going to show you how to do it in PowerPoint. So remember that we just acquired the 4 images. So I'm going to import them into PowerPoint. Okay, we have all 4 of them. So you can select all of them and you can make the size a little smaller so they all fit in one slide. And here are all your images. One thing I like to do is I like to arrange them next to each other, and I like to keep these gaps consistent so it looks nice and pretty. Okay, then what you need to do is you need to label them as A, B, C, and D, or you can actually write down what it is. So for example, you can do insert, and here you can write DAPI, for example. Okay, so we're gonna give it some color. So now everybody knows that this thing is DAPI. This thing is going to be our GFP, for example. Okay, and then you label the bottom two. And this will give you a very pretty figure. Now, in addition to having the figure itself, of course, you have to include the legend. The legend has to describe what it is I am seeing in this figure. And you have to do it to all of the images that you have acquired in the lab. Now that we have arranged these images side by side, let's have a look at them. They kind of look dim, don't they? We can make it look much better than this. So let's go back to ImageJ and open our image. Again, we're going to open it as a hyperstack. And what we're going to do now is we're going to split those channels. Okay, so we have phalloidin over here, we have GFP over here, and we have DAPI over here. One of the things we can do is we can actually play a little bit with the exposure. And when you play with exposure, it's important that you apply the same exposure parameters to all of your samples. So optimize it on one of the samples and then apply the same parameters to the other samples. So let's say we start with this. And when I talk about samples, I'm talking about individual samples, not different channels. So you should have the same parameters for channel, for example, 1 for the transfected cells and the non-transfected cells. So let's say we pick this image. So remember, when you work in ImageJ, you have to click on the image which you want to modify. So if you want to work on the second channel, you have to click on this. If you want to work on the third channel, you have to click on this. Now what I'm going to do is I'm going to go to Adjust, Brightness, and Contrast. Now, because we don't have too much time to understand exactly how it works, for now, what I would encourage you to do is just to click on Auto and see what this is going to do. Now, this looks awfully bright to me. I actually don't like the way it looks. So what I'm going to do is, this second slider is the maximum slider. Moving it up is actually going to make the picture a little dimmer. Now, I encourage you to read up on how exposure actually works and how these adjustment variables actually work. And I'm happy to discuss it offline as well. So let's say I like this image like this. Now the background here is actually not very black, so by moving the slider just a little bit higher, it will make the background a bit darker, which I really want. So these are your current parameter. Your image is now found between these two numbers, 517 and 790. This is for the third channel. Again, I encourage you to write it down and then use these parameters for the other images. All you have to do now is to click on Set. Okay, and this apply these parameters to this image. Now let's go to the C2 channel. This is your C2. As soon as you click on it, you'll see that these numbers are going to jump around. Oh, they didn't, but nonetheless, what we're going to do now is kind of repeat the same exercise. We're going to click on Auto, and what you're seeing now is that this is super, super bright. I actually don't like it that bright. So I'm going to take the slider a notch down, and this, on one hand, brings out that color, the green GFP that is over here. Again, this is kind of blurry, and that's because I didn't pay too much attention to when I was acquiring the image. Your images will look much, much nicer, especially after we clean all of the slides or cover slips. But anyways, this looks much better than this. So again, we do Auto, bring the slider up a little bit, just like that. Bring the minimum up bringing the minimum up will make the background much blacker, which is nice. And then we're going to click on Apply. So this image is done. And then we're going to click on the third one, and we're going to do Auto and see what this does. So you can see that right here there's enough dynamic range that the Auto actually doesn't do all that much. We're going to bring up the minimum just a little bit to make the black even blacker. and then we are going to make the images just a little bit, the white a little bit whiter, just like this. I like this. And then we're going to click on apply. And now we're going to go to image, and you can associate a lookup table with in each and every one of these images. So this is once again the blue one, the blue channel, this is the green channel, and the third one is your red channel. So you already know how to do that because I just showed you that. So all I'm going to do now is go to merge channels and merge these channels, just like that. And we are going to get this image. What I'm going to do next is I'm going to save this image again as JPEG, and I'm going to call it composite 2. And now I'm going to put it right next to this one to show you the difference between them. Okay, remember we said this is going to be 5. And now you're seeing these images side by side. Look at this difference. This is much prettier. It's much easier to see what's happening here. Okay, again, it's important that you understand that when you apply such exposure, new exposure parameters to your sample, when you modify this brightness and contrast, it's important to apply it to all of the images in this series. You can treat each channel separately, but all of the images have to be treated in the same way. So right away you can see this difference. Much easier to see what's happening here than this here. The next thing we're going to do is to use ImageJ to count the number of nuclei in our images. Of course, we all know that the microscopy can be used to generate these beautiful images, but we can use those images to also generate semi-quantitative data in software such as ImageJ. So let's open our image once again in ImageJ. So here's our image, the same image we've been working with for the past 20 minutes or so. The first thing we're going to do with this image is to identify which channel has our DAPI. And this is, of course, the first channel, C1 in this case. So this channel has all the DAPI. What we're going to do next is to go to Image, Color, and split the channels. We don't need the red channel, we don't need the green channel, we only need the blue channel. Okay. The next thing we're going to do is to duplicate this image. And the reason we're going to duplicate is because you remember I told you earlier, every time you do something in ImageJ, it modifies the image and you can't really go back anymore. But if you duplicate it, you don't need to go through the exercise of splitting your image every single time. So to duplicate the image, we're going to click on Ctrl-Shift-D. And that's going to bring this window, which essentially asks you how you want to label your duplicate. So we're just going to click on OK, and this is going to be our duplicate. So you see now you have 2 of them. So when I do this kind of analysis, I always like to keep my original intact. So this is my original, and I work on my duplicated file, and then if something goes wrong, I close it and I duplicate the original once again. So now we are ready to start with the analysis. Let's briefly start by just visually evaluating this image. Each one of these things is a nucleus. So we can see that there are lots of individual nuclei, but we also have some nuclei that are very close to each other. So we should anticipate that the algorithm might have some trouble distinguishing between these nuclei. So it might, for example, not be able to tell that these are 2 nuclei. It might actually count them as 1. So this is something we should be aware of from the get-go. So the first thing we're going to do with this image is to look at the brightness and contrast. And remember that you have to apply the same brightness and contrast settings to all of your images, all of the images that you're going to analyze. So let's open this window, and we're going to click on Auto just to see what it gives us. Not much of a difference. So let's bring this down a little bit. Again, I like to make this a little bit darker in the background. And I sort of like the way it looks right now. So I'm going to click on Apply. The next thing I want to do is close this window, and I want to identify all of the nuclei. Now, how do I identify all of the nuclei? Actually, just before I identify all of the nuclei, I'm going to convert this image to an 8-bit image. So you go to Image, Type, 8-bit. So now it's an 8-bit image. Now I want to identify all of those nuclei. What I'm going to do is go to Image, Adjust, and Threshold. So you can see now that all of the nuclei have been colored in red. What the threshold feature is going to do is going to convert the image into a binary image. Everything that is a nucleus is going to be white, and everything that's not a nucleus is going to be black. Okay, just a 0 and 1 kind of image, a binary image. Everything that's indicated here in red will be white. Everything that's indicated here in black will stay black. So basically what we need to do is to make sure that we've selected all of the nuclei. So you can see right now we're still missing some nuclei over here. So we can play with the sliders a little bit until we cover everything we want. Okay, so we're going to play with them until everything we want has been captured. So this is a little better. So you can see that all of these nuclei that were a little dimmer are now captured. You can also see it here. So when I play with the slider, you can see how it incorporates those nuclei. Now kind of looking around, making sure that our settings are perfect, or as perfect as they can be. This is good enough for now. What we're going to do now is click on Apply. This is going to convert the image into this binary image. Okay, so now we have a binary image. What we need to understand now about this image is that the white represents our nuclei. The black represents the background. The next thing we would like to do is to quantify the number of nuclei we have in this field of view. Now, you will remember that this nuclei, for example, were individual nuclei, but here we have two nuclei that touch each other, two or more nuclei that touch each other. Now, there are different ways how you can quantify nuclei in ImageJ. Some of them are very simple, and some methods are much more complicated. Now, I'm just going to introduce you to probably one of the simplest methods, and that's because it's the first time you're doing this kind of analysis. So now we have a binary image. If you go to Process and you go to Binary, it gives you different options that you can apply to your binary image, and I encourage you to play with the various options here. If you click on Watershed, however, you'll see that it introduces these lines. So you can read up on what exactly watersheding does, but from a practical perspective, you probably remember that we had two nuclei here, and now this line sort of separates the two nuclei into two nuclei. So it will no longer count this as a single big nucleus, instead it will count it as two nuclei. Of course, you can see that this particular watersheding function was not ideal, because you remember that here we had maybe 20 different nuclei, but for the time being it just counts this thing as one giant blob. And now we are almost ready to start counting the nuclei, or tell the machine to count the nuclei. But before we do this, what I would like to do is to estimate the average size of a nucleus. So let's pick a few of these nuclei. Maybe let's pick this one. So you select the circle or the oval selection marker over here, and you select, let's say, one of these nuclei. So like this. And now you click on Control M. And this opens this new window where it tells you that the area of this nucleus is 356. And you repeat it with just a few more nuclei. And you don't have to be supremely accurate here. Okay, so you pick a few more, maybe this one as well. This looks like a nice little nucleus. We're going to pick this one, sorry, right here, Control M. And maybe just a couple of smaller ones. Maybe this one right here looks like a nice and circular nucleus. Okay, so Control M. So you can see that the smaller ones are have an area of close to 200, and on average many of them are in the 300s. And there's some much bigger ones like this one, for example, which may or may not be more than a single nucleus. Okay, but we do know for a fact that these big blobs, are more than one nucleus. So let's estimate the size of this big blob, for example. So maybe something like this. Again, not supremely accurate here, but you can see that this thing is much, much bigger. So why do we need this information? First of all, make sure you record this information somewhere else, because the next few steps might actually erase this information. So we're going to remember that the smaller nuclei have an area of about 200. Now what we're going to do is we're going to click on this field anywhere just to remove this selection tool from here. We're going to go to Analyze and Analyze Particles. So now we're going to tell the machine to essentially start counting how many particles it sees in this field. Now, in terms of units, this area is measured in pixel square units. Okay, so you can see here that the image right now is represented in pixels, so it's not in millimeters or nanometers or anything like that. It's represented in pixels, and so the units are pixel square. And that's also important because sometimes these images are not in these units, so you have to convert the units. But in this case, it's all good. So now we need to tell the machine to start counting our particles. Here is our size. We're going to tell the machine what kind of sizes we want to include in our counts. So we want to exclude things that are too small. For example, this speck over here, we don't want to count it, or those little tiny things. We don't want to count those. And maybe we don't want to count these big things either, because we don't really know how many nuclei we have in those things. So we remember that the smallest thing was about 200 pixel squares. So let's start our counting maybe 150 pixel squares to really make sure we count all of these smaller nuclei. Now, things like this, for example, will be about 30 to 40 pixel square in size and therefore will not be included in this count. We're also going to set a maximum limit. So we know that this big thing was about 5,000 pixel square, so maybe our maximum should be somewhere around 2,500, so we don't count these big things. Okay. We are not going to touch circularity for now, and we're going to select all of these features. Okay. And all that's remaining to do is to click OK. The moment you click OK, it's going to ask you this whether you want to save the 7 measurements. These 7 measurements. We don't want to save them for now. And then it's going to open a new window. It's going to modify this image, and it's going to update this table. So let's see what exactly we are looking at here. And we're going to start with a summary. Actually, no, we're going to start with this image. In this image, it actually physically shows you which circles or which areas it actually counted. And each one of these areas is associated with a number. Okay, you'll see in a second what this number actually means. So now what you can do is you can actually put this right next to this one, and you can kind of estimate how good of a job your algorithm did. And then you can go back, duplicate this image, and go through all of these steps once again to adjust the algorithm to make sure it performs a little better. Once you have an algorithm that works well, you apply the same algorithm to the other images that you need to count, where you need to count the nuclei as well. You don't modify the algorithm between images if you want to compare the number of nuclei in multiple images. Okay, so we have all of these numbers. Let's look at what the summary table tells us. The summary table gives us the name of this image. So this is this image that we just analyzed. It counted 319 areas over here that fall within that range. Okay, so it counted 319 potential nuclei. And again, you can look at this and see how good of a job it actually did. Now, if you want to look at the individual nuclei, for example, you want to estimate how big this nuclei is, number 70, then you go to this table labeled as results. This gives you individual information about each and every one of these areas. So for example, we said we want to look at number 70. You go to number 70, it shows you that the area of number 70 is 797 pixels square. Okay. If you go, for example, to this number 177, remember it's this ones that we kind of selected to estimate how big the nuclei are. 177 is around 265 pixels square. Okay, so this kind of analysis allows you to very quickly count the number of nuclei in this field. After you have adjusted all of your parameters and you perform this kind of analysis for all of your images, you really have to summarize all of this data. So how do you do that? Well, let's open Excel over here, and let's say we captured this kind of data for 3 images. Let's say we did it for Gila, that were control, and we did the same kind of analysis for Gila that were inspected with GFP, and maybe we did the same thing for NIH cells that were control and NIH GFP. And then when we perform the counts, maybe we counted, so let's say for this one we counted 319 319 nuclei. For this one we counted, let's say 300 nuclei, and I didn't actually perform the count, so I'm just giving a random number just to show you how this is going to work. Let's say we counted 200 here, and let's say we counted 140 here. All you have to do now is to generate a bar graph. Okay, and you can generate a bar graph, any kind of bar graph that you want, but you really have to represent this data in an easy way for me to read. The next thing you have to do is you have to label both axi, you have to label everything in this graph, and you have to generate a legend. Okay, and this will give me an easy way to visualize the number of nuclei in your samples.